Hello, good morning and good afternoon. Welcome to the IQ Consortium webinar on current industry practices on closed system drug transfer devices for parenteral drug products. Before we get started today, I'm going to go through some quick housekeeping matters for you all. A friendly reminder that all participants will attend and remain on mute. Questions can be submitted throughout the webinar using the Q&A. But we will reserve 15 minutes at the end to address all questions received and take new questions as well. So please use the Q&A feature throughout the session, but know that we will pause at the end to address the questions that have been received and also welcome new questions. This session is being recorded and will also be made available after the webinar on our IQ Consortium YouTube channel. And so once again, we thank you all for joining us for this webinar on current industry practices on closed system drug transfer devices for parenteral drug products that has been organized by the IQ CSTD working group. And with that, I am pleased to introduce our first speaker. Sareen Jabari is a scientist at Biogen in the Drug Product Development Group, responsible for end-to-end drug product development from first in human to commercialization readiness. And with that, I'll turn it over to Sareen.
Hi, everyone. Good morning and good afternoon. Thank you for joining us today. My name is Sareen Jabari from the IQ CSTD Working Group. I am joined today by my colleagues, Isabella de Jong and Christian Lihamea, and together we will walk you through the key findings of our recently published manuscript. This manuscript was developed as part of a group effort, and we will be presenting it on behalf of the larger team. The IQ CSTD Working Group is made up of representatives from different biopharma companies, all coming together with a shared goal of better understanding and advancing current industry practices as it relates to CSTDs. Next slide. Next one. This wouldn't have been possible without the continued support of the IQ Consortium. The IQ Consortium is a technically focused, not-for-profit organization made up of nearly 40 biopharma companies. IQ advances innovative solutions to biomedical problems through science and technology. They augment the capability of member companies to bring transformational solutions that benefits patients, regulators, and the broader R&D community. The IQ Consortium is what brought this wonderful team of experts together. Next slide. To bring you both this manuscript and presentation that is about to be shared with you. I want to take a second to thank the team for all their hard work and all those all the times that we met and all the advances that we made to this manuscript. Just want to thank everyone on the team for that. And right down there is actually a snippet of our publication that you can find in the Journal of Pharmaceutical Sciences. Next slide. Today in our presentation, we're going to talk about how this came about with the USP 800, how its impact to CSTDs and the impact that it made to clinical site practices. From this, the challenges that it actually brought for drug manufacturers to have to navigate this new landscape. So together we developed a survey and we gathered insight from the industry, and that is what our manuscript covers. It's the outcome of those questionnaires. So we're going to share those findings with you, and then we'll wrap up and we'll share with you what our next steps are, and we'll open the floor to Q&A. So with that being said, Christian is going to kick off the presentation and tell us more about CSTDs.
Hello, my name is Christian Lihamea. I'm from Novartis, and I'm also part of this CSTD working group. Starting talking about CSTDs, there is no way around USP 800. So this USP 800 establishes basically first standards on safe handling of hazardous drugs in the healthcare setting, so minimizing exposure to healthcare professionals, to patients, and also the environment. And in there, CSTDs are mentioned as supplemental engineering controls when handling hazardous drugs. And there is two parts CSTDs are mentioned. One is the during compounding, they're actually recommended if the dosage form allows. And compounding, we're talking really confined areas, pharmacies with hoods and so on. So there is a lot of protection equipment available. So there they are recommended. On the other hand, for the administration part, so when the drug is compounded and leaves this pharmacy, there they are required in the administration area because yeah, there is not much other safety measures when it comes to protecting nursing stuff, for example. Then this USP 800 also refers to the CDC NIOSH list, so Center of Disease Control NIOSH list for hazardous drugs, that contains a list of what are hazardous drugs, basically. However, only commercial drugs. So what is not in this list are all investigational drugs. Next slide. So we talked about this abbreviation now a lot, so CSTDs. But where is this coming from? This is also from CDC NIOSH, where they described that such a closed system drug transfer device mechanically prohibits two things. One is the transfer of environmental contaminants into the system, and second is prohibiting the escape of hazardous drug, but also hazardous vapors from the drug out of the system. So a closed system. There is a multitude of systems out there, and the picture only really shows a very high level of a few CSTDs out there. But also during our survey, we identified it as common pharma companies there. We have like stumbled upon more than 30 different materials of construct that get into contact with our drugs. Next slide. So just to assure that that you know what we are talking about, I want to briefly explain to you very high level how CSTDs are used. So this is basically for the drug transfer from the vial and most of the time into an infusion bag. And there's two parts, so the compounding part and then the part where you are closer to the administration. For the compounding, you will first take this vial adapter, so here depicted in green. You will put it on top of the vial. Then you take your syringe and the syringe adapter, connect it to the vial. You then, step 3, withdraw, you disconnect. And this is the simplest, most simplest compounding step you can imagine. Then next is then the administration part. There, a third CSTD component gets added, which is this in red, the bag spike. So you will attach this bag spike to the infusion bag. And this bag spike has a side port that connects to your syringe adapter. So in step 2, you will connect. You will then in step 3 transfer your drug into the bag, and then you disconnect, and then your bag with this CSTD component attached, so to say, with this bag spike attached, is ready. Yeah, to start the infusion. And yeah, this is the very simple setup for compounding, and will be important in a second. Next slide. And when we discussed the survey results, what got imminent very fast is that, yeah, so there is a voluntary standardization using CSTDs out there in the clinical practice, and it's to basically reduce the exposure to potential hazardous drugs. There is also a part that these are needle free, so there is also no risk of needle piercing injuries, and because they fit for hazardous drugs, it's also a trend to implement them uniformly in site-wide policies, because it's a one-size-fits-all procedure, and meaning that we also got aware that they are then used for non-hazardous and as well as hazardous drugs, simply because one procedure is established and at sites. This standardization drives then that clinical sites might have 1 or 2 CSTDs available, and this causes then less training efforts, obviously, and also the inventory management is easier. there are regional differences, so regional different trends, because the more obviously there is a trend towards centralization, so big centralized pharmacies that compound and distribute this then to the different hospitals, the bigger, of course, the pressure to come up with a uniform procedure. Next slide. But of course, or although the protection of the healthcare worker is paramount, one has to also understand the other side. So CSTDs can have a potential impact on the quality and the safety, and ultimately then also on the efficacy of a drug in clinic, especially in these clinical studies, in early clinical studies. And there is these two parts, the correct dosing and the right quality. And I first want to focus on the right quality. There is a lot of literature published on this part. So there is a potential impact on protein particles, lubricant particles, stopper coring. There is transfer stress because there's small canals, the drug is pumped or moved through. There is also potential material incompatibilities. As I've mentioned, there is more than 30 plus materials of construct. The other part, the correct dosing, is really the core competency of clinical sites, but there is also quite some pitfalls in CSTDs that are maybe not always fully understood, and this is where I want to spend a little bit more time in the next two slides. So when it comes to correct dosing, there's two setups, and I start with the easier one first. So you want to completely transfer one dose from one vial to one patient. So you need to get your dose out and transfer this 100% to one patient. In this case, just a side note, CSTDs have a little bit of a higher non-extractable volume from the vial, so this A part, compared to a needle syringe extraction. So just USP 1151 overfills for 2 milliliters and down might not be sufficient, but that's not the point I want to make here. So if you manage to extract the right dose and you want to transfer everything, you basically would attach your syringe to the bag spike and you push your drug into the bag. But as you see here in this light blue color, there is a pathway staying with concentrated drug. How to get this transferred as well if you want 100% transfer. This would be, for example, performing this flush procedure. So you move back the plunger of the syringe and forth, and by that diluting out with additional volume from the bag this concentrated drug from these pathways. Attention, this has to be allowed by the CSTD brand. So this CSTD should allow for that, but also for the product, because this is additional mechanical stress. But if this is allowed, you can manage 100% transfer into the IV bag. Now, the other more complex part, and this is the next slide. Especially in early clinical studies or dose finding studies, when there is now an increasing trend of more potent drugs, ultra low concentrated drugs, where one ends up in the need of transferring very small doses in the beginning. Here, what will happen is that you connect your CSTD or your syringe to the vial, and when you withdraw your dose, you don't completely empty the vial, or if you pool, you don't usually completely empty the last vial. What will happen then? So in part C, so what you see in the syringe and the syringe adapter, you will basically withdraw additional invisible excess drug or excess volume, because this part is not visible on the graduation of the syringe and will be hiding in there. And if you now want to transfer your drug to the IV bag, you only have two options with CSTDs. One is an underdosing and the other one is an overdosing. With a simple flush procedure, I've not stumbled upon a solution how to 100% transfer. So what is going to happen? So you will attach the syringe and you will push this into the IV bag, and what then will happen is that what is in B, so the back spike, this will not be transferred. And with this, you will cause an underdosing. So for example, very small volumes, 0.5 milliliter. With CSTD1, you will underdose 33%. With CSTD2, you will underdose 43%. Um, on the other hand, if you then perform this flush, you will add this invisible excess volume to the bag as well. So what will then be the result is that you will overdose by 61% for CSTD1 or 20% for CSTD2. This sounds now very high, and yes, the volumes in this example are also very low. But this hopefully explains to you what potential pitfalls on these CSTDs are. Yeah, there is, I can also not give you a general number because this really depends then on the different CSTD brands, what the dead volume of this part C or part B really is for this specific brand. But the good news is if you start transferring 5 milliliters and up, you actually come somewhere into a range where the transfer error is below 5 to 10%. So yeah. So if you take one thing out of this webinar today, this is the part I hope you will remember. How we then, as pharma companies, navigate this complex environment of potential impact on quality, of trying to assure that the right doses are transferred and that the clinical trials are efficacious and safe. I would now like to hand back to Serene, who would bring us more through the survey and the different strategies that we came up with on covering this.
Mhm.
Thank you.
Thank you, Christian. So as Christian just explained, there are some challenges that we have seen as drug manufacturers. So in order to benchmark these challenges, we decided that as a team, we needed to get together and come up with a survey that helps address the different topics for the survey, so that way we can gather information and come to an industry consensus. So together we developed an anonymous 40-question survey, and the respondents were from the 15 global IQ Consortium members. The topics that were covered in this survey were the current strategies to perform CSTD compatibility evaluations, the management of CSTDs as they're used in the clinical sites and clinical studies, inclusion of all injectable drug products. So we're not just talking about biologics, but even small molecule, small protein molecules. And yes, we do for both protein and the small molecule based products. Next slide. So we issued out this survey, and we gathered back feedback, and from this is how we authored our manuscript. And so we're going to walk you through our key findings that I feel are impactful on industry level. Next slide. Okay. So first, you want to start off by navigating the clinical landscape. So what that means is that you want to preserve the drug sponsor clinical site relationship. So what this means is that you want to have early engagement with your stakeholders. Introduce a survey. We found that to be the most effective way to gather information on clinical site practices. In this survey, you should be able to gather how CSTDs are used at the site, if they're mandatory, are they used to prepare all drugs. Sometimes it's from a convenience perspective, as we mentioned earlier in the slides, that it just becomes a uniform approach to preparing drugs in a clinical setting. And this is highly favorable when the sites are centralized for the compounding. So that brings us to the understanding the scope in which the CSTDs are used. Sometimes they're just used for administration. They can also be used for compounding. So what you want to do while you're collecting this information from your clinical sites is be ready with a response. And what that looks like is that you want to communicate the risks and align that with your sites on the need for use of these CSTDs. If you have a non-hazardous drug, but your clinical site insists on using a CSTD, there needs to be a risk mitigation strategy to address this. And so what that means is that you can have a risk assessment, or you can supplement this with experimental testing, or a combination of both. In extreme cases, sponsors may disallow the use of CSTDs or even entirely discontinue the relationship with that particular site. That comes down to the information that you've gathered in your clinical site readiness and also understanding the mechanisms and the PQA profile of your drug. Next. Okay, so early communication, we just touched on that. So the next thing is having a phase appropriate strategy, right? So if you're early on in the development, you don't want to necessarily test 5, 6, 7 CSTDs. It takes a lot of resources, a lot of time. So the selection of your CSTD evaluation will essentially depend on that initial communication that you have with your clinical sites, and knowing what their available inventory is and the known compatibility with the materials of construction and your pipeline products. And during the early phase, most companies evaluate between 1 to 3 CSTDs, but the author recommends between 1 to 2. Now, as you move later into development and closer to your commercial launch, the testing is often driven by the clinical site request. At this point, you have a deeper understanding of your mechanism of action, your drug interaction with the materials of compatibility, your dosing regimen. And so for these things, you have a little bit more, a broader understanding of what your drug will how it will perform with different CSTDs on a global scale. There are CSTDs that are available in other countries that are not necessarily available here in the United States. And so that's something that should be kept in mind as you go through the development process. So a couple approaches that were recommended in the manuscript was having a risk assessment and experimental testing. Next slide. So what is this? What is the risk assessment? What should you include in there? So risk assessment isn't necessarily done as a one-time thing. It can be done at any time during the course of your development and the course of the phases that your drug will move into. These risk assessments can be a decision tree, a flow diagram on company strategies and the drug candidates. Evaluate the risks of the CSTDs with the drug PQAs by understanding the physical and biochemical stability of your drug with the components of the CSTD that it will come in contact with. Also, can your clinical preparation volumes be administered 100% dose without underdosing or overdosing? Other things that you should consider in your risk assessments are your formulation and the surfactant levels. The hazard class is very important. If you know you have a hazardous drug, so you're definitely having to use a CSTD to prepare the drug. So all of this comes into play onto how you want to qualify CSTDs, how you want to evaluate them. So the challenges, as we mentioned, are included in the low product concentration and dose volumes, where you can have a potential to lose a good chunk of your drug that you were planning to dose. So it's a good idea to leverage the knowledge of CSTD compatibility, have an understanding on the holdup volumes, different viscosity. Drugs will have different holdup volumes in these devices. And also understanding the interactions with your materials. Some of these CSTDs, they shed particles. So that's there's strategies to prevent that from happening, but it's just something that when you start talking about experimental testing and leveraging the knowledge of your interaction between your drug and your materials of compatibility, that's a big one that we saw. And it's like a repetitive pattern. But Isabella is going to walk you through the risk assessment, the experimental testing and the other strategies in our manuscript.
Thank you, Serene. My name is Isabella DeYoung. I'm in the pharmaceutical development at Genentech. So I'm going to discuss now the analytical methods and evaluations. We asked, as part of developing this paper in our survey, what are the methods analytically that are typically used when experimental testing of CSTDs is performed. And respondents indicated that the top methods included protein content measurement, appearance for clarity and opalescence, subvisible particles, protein purity by SEH-PLC, dose accuracy and recovery, and hold-up volume and appearance for visible particles. So a few respondents also indicated that they also use microflow imaging as additional characterization of subvisible particles. And this is a useful method for helping to differentiate lubricants for other particles that might be detected. And then looking at what is done through stages of development, protein content, appearance, and subvisible particles. These methods are all used more frequently during early stage, but as we move through development, all these methods are applied as well as additional methods. Next slide, please. When these methods are applied during experimental testing, issues identified by the respondents include subvisible particles, appearance and visible particles, hold-up volume, specifically extractable volume, dose accuracy and recovery, SEH-PLC, and flow imaging microscopy. And again, this is similar, these observations through analytical testing as you move through early, late, and commercial evaluations. It's the recommendation when testing for product quality impact to select analytical procedures based on the specific product. As Serene indicated before, you have to look at your product liabilities, indication, and patient population, and what are the critical attributes that you think are going to be important. Look at experience that you may have, in particular with other in-use studies that you may have performed, and also follow CSTD vendor instructions when you're doing your experimental testing. Next slide, please. So when studies are done, when you've done your risk assessment, as indicated before, it is important you may have to do these experimental tests. A lot of companies indicated that they may do a more limited number of CSTDs for early development. This is typically because studies are smaller with a more limited number of clinical sites. And if you've been communicating with your clinical sites, you may be able to find out in advance what CSTDs they'll be required to use. As you move through development, particularly for late or commercial phase, you may need to add additional CSTDs to test to support a broader number of clinical sites, or if you're moving to more global clinical study. In cases where issues are identified, if the risk is too high, it may be necessary to disallow CSTDs. Respondents indicated that in cases that issues are identified, they've implemented some mitigation strategies in order to be able to support the CSTDs. For example, a 0.2 micron inline filter can be applied if particles are detected or if there's concern about particle risk. Of course, you'll have to have confirmed that your drug is compatible with an inline filter before that can be implemented. Some companies indicated that in case there are issues with extractable volume, they've been able to allow sites to use an additional IMP vial. That, of course, is something that you'll need to confirm based on your actual clinical supply. And then finally, flushing of CSTD components may also be applied, but as indicated, it's important to read the instructions and understand the implications for over or underdosing with CSTD flushing. Just want to emphasize again, proactive communication with clinical sites is very important. Ask about the required components for preparation and in administration. Collaborating with clinical sites ensures a smooth clinical trial execution. When we asked the companies we surveyed what they do in order to decide what CSTDs to test, most respondents indicated that they did pre-study surveys with clinical sites to understand what they're using and what their practices are. Sites then indicated they'll do experimental testing for compatibility or take a risk-based approach or negotiations with clinical sites. Next slide, please. When communicating with clinical sites, one of the best tools you'll have besides pre-site surveys is the pharmacy manual. Any instructions or guidance or company position about the use of CSTDs should be proactively included in the pharmacy manual or in the drug preparation instructions of the pharmacy manual. In some cases, clinical sites will have a policy that CSTDs are required regardless of hazard drug status. Companies have indicated they've been required to provide evidence of incompatibility in order to waive the site's internal procedures. In some cases, drug manufacturers have permitted the use of administration components in order to allow the protection of the nursing staff, since this is a requirement, but have been able to have sites avoid the use during the preparation by pharmacy staff. Additionally, some sites, based on these discussions and feedback from clinical sites, have done initial testing or found ways for acceptable usage of a CSTD that may previously have been disallowed. And this is where, again, really understanding the risk and the issue and understanding how you can allow it if you really want to enable a clinical site. In some cases, sites have indicated when they cannot implement a CSTD according to their procedures, they may opt to withdraw from a clinical study, or a drug manufacturer has had to remove a clinical site from a clinical study. Next slide, please. In regard to communicating to the health authorities or reporting to health authorities, most respondents indicated that they don't include details of CSTDs, the MOC, the material of construction, or the brand names in regulatory submissions, such as the CTA, an NDA, or commercial label. Typically, commercial drug product manufacturers don't specify CSTD compatibility on a commercial label. Though we did find one example of a company that did include some language about file adapters and the MOCs associated with that on their label. In regard to co-packaging with a drug product, in this case, the details of the CSTD would be required to include as part of the submission documents. Under FDA 21 CFR 3.2, this would become a combination product. Next slide, please. When CSTDs are used by healthcare providers, practices relating to the monitoring of the CSTDs were evaluated. Most respondents indicated during early clinical stage, there's a very increased concern about dose accuracy during entry into dose escalation studies. And where the safety profile isn't well understood, there may be additional monitoring. By the time we get to the commercial phase, we move through late stage to commercial, there tends to be less concern as the safety profiles and understanding of the product is more well defined. There could potentially be efficacy risk if a full dose is not being delivered, so it's important to be mindful of that risk. More than 90% of surveyed drug manufacturers don't have a mechanism in place to receive information about changes made to the material of construction or the manufacturing process of CSTDs used with clinical trials or with commercial drug products. And when we asked commercial sites if they're monitoring or evaluating, most respondents indicate that they do collect the CSTD information at clinical sites for an investigational drug. However, as the clinical study proceeds, they don't necessarily maintain monitoring changes during that study. Next slide, please. And so I'd like to hand it back to Serene at this point.
Thank you, Isabella. So in summary, drug manufacturers are utilizing various approaches to evaluate CSTDs, as you have seen throughout the discussions in our slide. There's a strong emphasis on communication and early engagement from the sponsor with the clinical sites. And to understand the clinical site landscape, understanding the site practices and how that relates to CSTDs. Convey the drug product manufacturer's position on CSTDs, on whether you don't want them used at all, used in some cases, where some are allowed, disallowed. Use phase appropriate risk mitigation strategies. You know, as we discussed, risk assessments, they'll probably be your best friend. In experimental testing as needed to supplement the information from your risk assessments, or maybe your risk assessments could drive to that solution of you needing experimental testing. Communicate clear handling instructions in the pharmacy manual. Ensure that you're on the same page with the people that are preparing these medications to deliver to patients. And also being mindful of the changing global landscape. And there's ways to do that by remaining involved in the industry consortia, papers and webinars such as this one. And you want to also monitor the evolving agency expectations to ensure that there's alignment with emerging standardization for CSTD evaluations, and in order for proper validations of your dose delivery. Next. So that brings us to what's next, right? So we published this manuscript, we communicated what the industry stance is, what our practices are. So what we have going on now is that we have a white paper that's in progress. We're authoring this to have a standardized way of testing CSTDs. This is currently under review and we're expected to publish it later on this year. And also USP 800 is expected to open for public comment sometime this year. So we're preparing to align responses so that way we are ready for the next revision. So with that, I would like to thank you all for your attention to our presentation, and I'd like to open it up for Q&A. Feel free to use the chat on the side to pop in your questions. Okay, while you're generating those, I have one for the panel. Let's see. Okay. If this manuscript were to be expanded into future work, what questions remain unanswered?
I think regarding the communication part and finding out and how to work with clinical sites to cover these risks, I think we have reached quite a good status. I mean, it depends on what's coming next. So currently, I feel it has reached kind of a steady state. So the field on CSTDs has not moved so much in the last 1 to 2 years.
Mm-hmm.
But I mean, there is probably the next CSTD coming out.
Yeah.
Kristen, I'd like to add, I think it would be great to really understand, while, we haven't, we're in a nice place. I think the continued education of healthcare providers and ways of doing that, I think really working in the future with regulators as well, so they understand the potential risk. And I would love to see in the future kind of a pulse check in another few years. what have been the healthcare provider practices that have changed. You know, when we started this, looking at this years ago, there really wasn't a lot of education or understanding, but we've learned so much about the way healthcare providers are using CSTDs, what they're up against in terms of implementing CSTDs and the training at clinical sites and healthcare providers. And I'd love to learn more about that in the future. Where has the needle moved in the hands of the healthcare providers?
Mhm. That actually brings us to one of our panel questions. It says, what are next steps regarding an interaction with regulators? That's a great question. Our next step is actually the USP 800. And in conjunction with the publications that we continue to produce, getting their attention in that way will help influence and shape how the practices are going to be done in the future.
Yeah. So regarding this next publication, what we were thinking is in order to be able to compare different CSTDs, one somehow has to define a standard, how to test them, define a surrogate, not to be linked to specific drug products that you might have to source from the market very expensively. So I think with having or working on a protocol, how to test CSTDs and be in the end able to compare them is something like the next step. And I mean, maybe I don't know what will happen and what will regulators might do with such standards, but yeah, we can try.
Yes, yes. Our next question is, do you have a gauge of how common co-packaging has been to date? Well, there's only one case I think we have of, it's not necessarily co-packaging, it's that
Yeah.
they mentioned that a adapter is used, right, as part of the drug delivery. And that was a screenshot in a previous slide.
Well, yeah, just to clarify, they didn't indicate that it was co-pack. They just mentioned that when a vial adapter is used, those are the materials of construction. In terms of actually co-packaging and commercializing a drug with a CSTD, I'm not aware. I haven't been alerted of this or what's going on in the industry, so I don't know if anybody else has heard of this happening. I think.
Yeah, I'm also not aware of such a case. The thing is, as we've mentioned, there is a big need also for standardization training and alignment in hospitals. So if you come with your CSTD, how to assure that this can be properly used. So even if it's co-packed, what help I don't know, the chances are probably not so low that they just take it and throw it away and use their CSTD. So that's the co-packing is probably not the solution. That's what I want to make.
Yeah, agreed. So we have a good question here. It says, would you please expand on strategies to overcome underdosing, overdosing at low volumes, less than 1 ml? Our recommendation would be not to use a CSTD if you can avoid it for low doses.
I mean,
it's very hard to retrieve.
There is very complex other flush procedures, so you can always then use a fresh syringe and the syringe adapter filled with with your IV diluent and then flush this out of the back spike. So there is methods, I would say, but they are not routine. So I think this is something you would really then need to clarify in a one-on-one discussion how to do that.
Yeah, there's a nice paper by Grzinski. I think we referenced it in our paper that also talks about the risks and the different flush procedure in terms of your accuracy and your risk of flushing. I think that's the paper, and I know that you've done a lot of work in Kristen. So I think it is very challenging because of the holdup volume, but very, very careful work to understand if flush procedures can be used.
have another one here. So it says, do you see an issue with reconstitution of freeze-dried products using CSTDs? In our evaluation with the high holdup volume, we see it as a risky approach to use CSTDs for reconstitution and withdrawal of freeze-dried vial products. What is your opinion?
There is an additional step where you can have holdup volume when you add your diluent to the vial. There is also flush procedures described. I don't know if it's in this paper you've mentioned, Isabella, but there is also sometimes air pushes described. So you fill in air and try really to push through the remaining liquid from the vial adapter into your lyo product. Yeah, things like this. But of course, with reconstitution, there comes an additional step with CSTDs used.
Yeah, there's potential additional for variability when introducing your reconstitution diluent. There's an additional risk of introducing particles. Again, I think you have to read the instructions. I think a lot of CSTD manufacturers do provide instructions on this and follow the instructions and test it and understand what the risk is and if you can still reconstitute accurately and then also remove the dose. But it's also going to depend on the total volume of the vial and your cake and how much you need to do that reconstitution.
Mm-hmm.
So there's another question. Do you see no, wait, a question to me, when manipulating small volumes, so around 0.5 ml with CSTDs, what would be your best recommendation to reach appropriate target concentration? So there is, except from what we already described, there is one other potential is you dilute it even more. So if you can increase the volume, then these void volume topics and non-extractable volume issues are reduced. So if you can make out of 0.5 ml 5 ml, it gets much easier to handle, obviously.
Mm-hmm. We have a lot of questions here. Are there experiences about the willingness of CSTD manufacturers to agree on change notification related to materials, dead volume, etc.?
I think
we're working on it. Yeah.
Very dependent on the CSTD manufacturer. However, one of the challenges is that in the experience and what's been cited to me is that they're not typically willing to provide this information unless you're co-packaging. If you want that information, you have to, purchase a certain volume and co-packaging with your drug product. Then there's a lot of willingness to work with drug manufacturers. But if you're not in a co-packaging situation, it can be challenging to have those agreements implemented.
As we have here, so if the CSTD remains attached to a vial for multiple withdrawals, what is the validated in-use stability and sterility window for that specific formulation device combination?
I think you'd have to understand that for your formulation and the device. There's a lot of guidance in the USP, but it's important to consider the formulation, the device, but you also have to understand what the potential microbial risk and how that's going to be used. So I don't know that we can tell you what it is. You have to look at the guidance and understand for your product, for your formulation and the device in question.
Mm-hmm. Thank you, Isabella. So there's if the sites want to use a different CSTD than qualified by you and the materials and residual volumes are comparable, do you confirm? That kind of goes back down to your risk assessment and your need for experimental testing. Because if you already have demonstrated material compatibility with the materials, you don't necessarily know if different silicone coating on the inside or like any kind of lubricants will leach off into your product. So these are things that would probably have to be covered in the evaluation, whether you do risk assessments or experimental testing. Let's see.
I've just tried to add a reference and I've lost a little bit track of questions already answered. Where are
It's
we?
okay.
I think
Oh, go ahead, Isabella.
no, I was just gonna say, I think the next question is, has the consortium considered a clinical site panel for feedback? I mean, I think that's a great idea. I don't know that if we've done this through the CSTD working group, but that could be a good idea.
What are the highest value aspects of USP 800 that the team wants to influence? We want to influence all of it.
I mean,
Yeah.
if it fits somewhere, the risk of overdosing, not only underdosing into this, because there seems to be really not much awareness about this. Yeah. So if this could be somewhere in a USP, but also there, I'm not sure if this will reach the clinical sites. If it's in a USP, I don't know.
There's another one here. Have you seen any issues with the lubricant oil used in these devices, such as incompatibilities and/or particles?
Yes.
Yes.
There have been a few publications that have talked about this. I think, sorry, and I'm not remembering the exact names. But there have been a few publications in 2020 and 2019 that discussed about these specific incompatibilities. They are referenced in our paper too, so you can look at those references there.
Is allowing use of untested CSTD based on a previous set of approved fluid path materials of construction an acceptable form of risk assessment?
I mean, there is several parts. So the compatibility part. So if the drug is compatible with a material, I think this can be assessed. The thing is, the volume. So if you can transfer the correct volume as well. So this part you would need probably to assess separately. It's not only the material. You missed one above. Have you considered partnering with CSTD manufacturers to test their products further with different drugs or substances?
No, we haven't. That's the short answer. But what we're trying to do is that through our publication, have it so that way the CSCDs are standardized to a set of testing. So that way, when we get it, we have some of the information has already been evaluated by the manufacturer themselves.
I don't know, Isabella, if you want to add something to that, or
Yeah, I mean, there's a group, the AAMI group, who has been doing some work with drug manufacturers in collaboration with CSTD manufacturers to publish a consensus paper discussing what the testing requirements are and what the challenges with CSTDs and risks are. So that's something though that's being done by a different group, the AAMI group.
Question here says, we have requested clinical sites to reconstitute lyovials with needle and syringe to have the correct starting concentration, and then use a CSTD to remove and transfer the drug product. Is this acceptable in your opinion, where there are two different puncture sizes on the vial stopper? Do you see coring? Do you see coring in your vial? Because that would be a good indicator on whether the stopper can handle multiple puncture sizes.
maybe one part, but the other one, I mean, it's not about sterility, because I mean, yes, CSTDs are a closed system, but you open them and then you attach them to your vial. So at this step, there might still be an instability coming into a vial. So not necessarily, I would say, a CSTD prolongs your hold time. So you breach the vial with adding the CSTD. So maybe this is the part. Also part of the question. So when you punch it a second time, as long as you're staying within your hold times, I think it's from microbial point of view, okay.
I have one more question. Oops. Oh,
I think the question is, have you included CSTD info such as testing and compatibility in your regulatory submissions? That was one of the questions that we discussed, and most companies indicated that they're not including that detail. However, if there's reasons why you think it's important to communicate this to the health authorities, you know, I think that's up to you to discuss if it's important and would be of value for your study to include that. We are not, I have not been made aware of this being requested, but I don't know what the experience is of other panelists.
Yeah, I've here also limited experience, but I mean, if it's a hazardous drug, and now with USP being referenced in other chapters of of USP, yeah, you probably have to have CSTDs enabled. So I can expect that there might be a question coming.
And certainly if you're planning on having a co-pack of a CSTD, then and you're going to commercialize that, then that of course would be a requirement.
mhm.
2 more minutes. Do we have,
Yeah.
or right? Any
Yeah.
unanswered questions? I know, Jillian.
Well, yeah, there was one about someone was asking about the reference for the paper where they can find it. It's the Journal of Pharmaceutical Science.
I added reference to this paper, so the survey. I don't know if this was the question. So yeah.
Mm-hmm. And is referenced in our slides as well. One thing I did want to mention, you know, this work, like I mentioned earlier in the slides, was done with the cooperation of 15 companies that are part of the IQ Consortium. It would be nice to expand on that number and have more people join our group. So that way we get, you know, a little bit more experience, a little bit more knowledge on what other companies that are outside the IQ Consortium, how are they navigating their CSTD development. So we encourage you to join our group. Are there any other questions or anything that we missed?
I think most is answered.
Okay.
You know, Jillian, you want to come on camera and close, or how do we do that now?
Yeah, thanks everyone, and we hope you found it informative. As we mentioned at the beginning of the session, we will be posting the recorded webinar on the IQ Consortium's website. Specifically, we have a YouTube channel that you all can access. And also, if you're interested, we will probably post a LinkedIn of this as well. So please check out our IQ Consortium LinkedIn page. That's another way to keep tabs on this working group as well as others' activities within the IQ Consortium. And with that, I think we'll close for today. Thank you to all of our wonderful speakers and to you all, our attendees.
Thank you.
Much. Bye.
Bye.